It is amazing having you here online, and it's such a pleasure and a privilege to be in your company with so much information. And having you in the Congress is going to be awesome. So we wanted to do a little teaser and just let our colleagues know what they are getting into if they come to Lisbon or have the online Congress, which is going to be awesome too. So just to give a little background, and correct me if I'm wrong. So Paul is a co-founder and chief executive officer of HairClone. And this is a company which is cell-based, cell therapy-based, and it's developing cell-based treatments for androgenic alopecia, which is awesome, which is actually, I think it's the future. And he has an impressive background, and he was a lead inventor on patents for multi-cell therapy and for treating chronic ulcers. And he also was the sole founder of Intercytex Limited, where there were developed different medications, including one for correcting scar contractures and dystrophic epidermolysis bullosa. I hope I said it correctly. And it also carried one of the world's first cell therapy clinical trial to induce new hair formation, which is huge in our specialty. You also have sat in a variety of panels, grant funding bodies, regulatory committees. You're a co-editor of the Journal of Regenerative Medicine, co-director of the Doctoral Training Center in Regenerative Medicine at Manchester University, and industrial director of Regenerate. So imagine, this is an impressive background. So my first question is very related to that. How did you end up in regenerative medicine? And then how did this evolve into regenerative therapies specifically for hair?
I originally was a collagen biochemist and worked for a small biotech in Boston in the US, developing a skin replacement. We developed Apligraf, which was the world's first regenerative medicine, which was approved by the FDA. And as you mentioned, it's being used to treat chronic ulcers. It's treated over 2 million people now in the US. And whilst I was at Organogenesis, my interest moved from collagen, the protein, to the cells involved in making the skin. And it kind of follows the evolution of pharmaceuticals. They started with small molecules, moved to biological molecules, and then now just beginning to move to cells themselves. And the important thing, as far as I'm concerned, with cells is they're responsive to their environment. They're not just like a drug in which you inject it or ingest it, and that's it. It works or it doesn't. Cells will respond to their environment, and more importantly for hair regeneration, they build tissues. So my interest since then, subsequently, has been in the cell side of regenerative medicine. And as you mentioned, I started Intercytex and worked on regenerating hair follicles. That was the idea. There was following on from things like papers like Colin Yehoda, who showed that the dermal papilla cells could induce new hair formation in mice. And that was really the start of the interest in that for hair regenerative therapy.
Yes, Paul.
Well, and then my interest then was in hair. And I think, as everybody in the audience will probably agree, hair is an amazing, fascinating mini organ. Most people don't really appreciate the full structure of it. And my interest for regenerative medicine, it's one of the only tissues in the human body that naturally regenerates in a cycle, the hair cycle of anagen, telogen, catagen. So the hair itself is a regenerative organ. And the interest there is in our ability to modify the hair's own inherent regenerative capacity.
That's awesome. Yeah, it is something that not only as physicians that are treating hair loss are interested, but that's a common interest around the world. And I know that it hit the news recently that hair clone was coming here, and cloning and hair cloning keeps on sounding in the background of every chat where patients consult. So when these patients come to the office, they love asking about when is cloning happening? When are you bringing it here? I know that some people are doing it or not, and perhaps not knowing the spectrum this involves, you know? So it would be awesome if you could explain a little bit of the difference between cloning hair follicles, rejuvenating hair follicles, and regeneration of these same hair follicles, because I understand there's difference between these terms.
Yeah, and it's probably different people have different definitions. My own personal one is hair cloning is when people have tried it in the past, splitting a hair follicle. So you get 2 or maybe at most 3 hair follicles per hair follicle explanted. There are various groups working on that. There's been some success. It's been variable. Hair regeneration is kind of the human version of what Colin Yehoda was showing. We implant some cells and they seed a new hair follicle to form, and a brand new hair follicle forms. We tried that in Intercytex. It was successful to some degree, and we did form new hair follicles, but they were new vellus hairs, which for a cosmetic point of view is not very helpful. But what we found at Intercytex and what people at Adoran's research found when trying to do the same thing, regenerating a brand new hair follicle, was that the cells that were implanted were rejuvenating, miniaturizing hair follicles. And that's a separate aspect. And I think way more possible from a clinical point of view, because it's a natural hair follicle. It's interesting because you could potentially convert the DHT sensitive hair follicle into a DHT insensitive hair follicle. But from a kind of hair transplant clinical point of view, much more interesting as a treatment, because instead of hair transplantation or hair regeneration, which would take someone with hair loss and a bald scalp, and then creating a new hairline, and then it's a kind of 1 or 2 hair transplants in life, and then the patient, that's the end of the interaction between the clinician and the patient. In rejuvenation's case, because miniaturization occurs as a wave of miniaturization, you could develop a lifelong interaction with a patient. Patient comes to a clinic just beginning to be concerned about hair miniaturization and hair loss, and you could be through life applying cells to that patient, rejuvenating the hair, rebuilding it, and the patient maintains their appearance, their youthful appearance, instead of going from an appearance of hair loss and then post-transplantation, which for some people can be traumatic. You are maintaining the appearance through life, and no one would know except the clinician and the patient that patient was actually going to lose hair had they not been treated.
That's nice. It's also good to know that I'm not gonna lose my job because I won't be doing surgery. I'm just gonna grow my practice. That's good to know. Exactly. Oh, sorry. So I know this sounds beautiful and it sounds promising, but I would love to know a little bit, just don't give us a whole cake, just a little piece of the cake of what evidence do you have that this rejuvenation and regeneration are in the androgenic alopecia at least has gone so far. And are you investigating only on androgenic alopecia, or are you seeing all these other types of alopecia?
Okay. We're concentrating on androgenic alopecia at the moment. I have found in the past, in running other biotechs, if you try and spread your targets, you reduce your capabilities, and you diffuse the amount of effort you have. Much better to do one at a time. So an androgenic alopecia is a huge clinical need and a huge market. So we're going for a large one first. Evidence of regeneration, and most of the studies have shown that mice are not the same as men. Regenerating brand new hair follicles is extremely easy in a mouse, and extremely difficult in humans. Probably the best evidence of this is the work of Colin Yehoda through his career. He has published numerous papers on regenerating new hair follicles in rodents, and has moved into the humans with some success, very limited success, but some success. The concern from my point of view, running a biotech company for several years, is the ultimate cost of that therapy. You would have to have something that was clinically very acceptable. You know, the correct thickness, the correct angle, the correct character. So you would have to be pretty good in your manufacture. You'd have to be reproducible. You'd need to make thousands of these hair follicles the same. And the cost of that, I think, will be extremely high. Evidence for rejuvenation, papers from Kevin McElwee had shown that in mice. There's evidence from Intercytex and from Adoran's research and from Replicell, all indicating that rejuvenation is achievable. So that there is more human data supporting rejuvenation than regeneration. And rebuilding, the patient's own follicles is going to be a lot more cost effective than regenerating brand new hair follicles.
That sounds very, very promising. And just without burning too much information also, are we involving cell expansion in this rejuvenation? And if so, what would be the source of those stem cells for expanding these cells for rejuvenation?
Yeah. Well, the cell of interest is the dermal papilla cell. And some people refer to these as stem cells. I don't consider them stem cells. They're not immortal. They don't grow forever. They have been shown to differentiate into other tissues, which is what people have been used to define them as a stem cell. But I don't think they are stem cells. But that's a little pedantic. The cells themselves, people are looking for different sources of cells. There's groups looking at induced pluripotential stem cell technology to produce the dermal papilla cell. At Hair Clone, we're just looking to take DHT insensitive dermal papilla cells from the patient and expand those in culture. We can expand them several hundred, up to about 1,000-fold in culture. So from a few thousand cells you can get from one hair follicle, we can make millions of dermal papilla cells in 2 or 3 weeks. They grow easily. The challenge is to do this under GMP. It's a highly regulated procedure, and we have to do them in clean rooms under good manufacturing practice. And we are at the stage at the moment of doing tech transfer of the lab-based system into the clean room-based system.
That's nice. So I know the process from bench to bedside, it's quite expensive, and it is very difficult to establish a specific availability date. But is there a timeline that we can share as physicians to our patients that keep asking about cloning, and we can tell them, okay, it's not cloning per se. We're going to expand some cells that we can rejuvenate your hair grafts and your follicles. We can actually rejuvenate them and make them unsensitive to DHT. But when they ask us, okay, I'm ready, when can this be available? We keep on putting deadlines like next 5 years, next 10 years. What would be like a certain date that we can share with our patients so far if this technology gets to our hands?
Yes, next year.
That's soon?
Yes.
I was
So
expecting like a 5 year at least. That
yeah,
sounds
no,
awesome.
yes. Well, there's a but. As I mentioned, I've been involved in cell therapy and regenerative medicine for many years, and are involved in the regulatory system for regenerative medicine. And there's a certain provision in the UK, and it's UK only, regulatory system that enables a clinician to prescribe a unlicensed treatment. So it can't be advertised. You can't make medical claims, obviously, but a clinician can prescribe it. So what we're planning to do early next year is to offer a cell expansion service to specific clinicians that we are in our clinical partner network. They could use that to treat patients. And one of the problems of doing a clinical trial is, in order to do a clinical trial, you have to have a very prescriptive protocol, which for a surgical procedure is very difficult to describe. And what we've found in the past is, in a clinical trial, you choose the wrong patients, you use the wrong inclusion-exclusion criteria, you may inject too many cells, not enough cells, too deep, too shallow. There's all variations. So by offering a cell expansion service, we would leave it up to the clinicians to use their skill to try it on patients, and to modify the protocol they use. As soon as we see clinical efficacy, we can use that then to inform the design of a clinical trial. And we would then use those clinical trials in the US, the UK, Europe, again, with our clinical partners. But we would know now that it works because we have been using it and it works in the UK. It sounds an unusual system, and it's unique as far as we're concerned. I've checked with the regulators to ensure that they're comfortable with this, and they're very supportive of it, because they say they don't like to see clinical trials fail, and for a clinical trial to fail for the wrong reason. So to have it as an iterative trial in a way, to go baby steps one at a time to develop a system that actually works. And then we can use that in a clinical trial, and we can use that then to gain marketing licenses so that clinicians around the world can use it. But patients could have access to it next year.
Wow. I can't wait to actually meet you and get some more questions with you on a one-on-one.
Yeah, you're very welcome.
And so thank you so much for your time. I know that scheduling, it's been hard, me being so far away from you, but thank you for sharing all this with us. And we definitely hope to see you soon. And
yes, I'm looking forward to
talk a lot more. I'm interested about that partnering thing. I want to be in the loop.
Yeah, definitely.
So thank you very much. And thank you everyone for being here and listening to this. Stay tuned, because this is going to be an awesome talk.
I look forward to it. See you in October.
Yes.